To set up a plate for the Miles and Misra technique of plating to count for colony-forming units, you need to first divide an agar plate into sections, each section for a different dilution as shown here. In this example, 5 dilutions can be plated out onto one plate. In each section, write down the dilution value for the culture that will be placed there. Using good aseptic technique, pipette out the first dilution of bacteria into the correct section of the plate. Add 3 separate 10 microliter spots for this dilution. Then move on to pipette out the next dilution, adding 3 separate spots of 10 microliters for this dilution as well. Continue to do this for each dilution until you have added 3 10 microliter spots of bacterial culture to the plate in the correct sections for each dilution required. Allow the spots of culture to dry before turning the plate upside down for incubation overnight. There are 2 ways that you can leave your plates to dry. If time is not an issue, you can place the lid fully on the plate and leave it to dry on the bench. If you need the spots to dry a little faster, you can arrange your plates as shown here under the protection of a blue flame with the lid slightly ajar. After overnight incubation, you will see that the spots of the culture have grown into visible bacterial growth. The lower dilution spots will have more colonies within them, so much so that they may appear to merge together and be very difficult to count. You should still be able to see where each spot was placed by the spread of the colonies formed. Count colonies in only the dilution that has between 3 and 30 colonies per spot, as it will be easier to count the colonies at this dilution, and errors in counting will be reduced at this dilution due to the fewer merged colonies that will be seen at higher cell concentrations, or too few colonies to count, as would be seen at lower cell concentrations. Now that you have chosen the dilution that you will count, you need to find the number of colony-forming units, or CFU, per mil in the original culture. To do this, count the colonies in your chosen dilution for each spot and take an average of these to find the average number of colonies per spot. Per spot is the same as per 10 microliters, as this is the volume of diluted culture that you plated out. So if, for example, you obtained an average of 5 colonies per spot, that is the same as 5 colonies per 10 microliters of diluted culture. If you counted an average of 5 colonies per 10 microliter spot at the 10 to the minus 2 dilution, you now need to convert that to CFU, or colony forming units per mil, in the original bacterial culture. First, you should take into account the dilution. For example, if you've counted 5 colonies at 10 to the minus 2 dilution, you need to times your number of colonies counted by 10 to the power of 2, or 100, to achieve the number of CFU per 10 microliters of the original culture. In this example, you will find 500 colonies per 10 microliters. Next, you have to convert this figure to CFU, or colony forming units per mil. To do this, we know that 10 microliters is 100 times less than 1 mil, so we times our number of colonies, in this case 500, by 100 to obtain the value of CFU per mil in the original culture. In this example, the answer is 50,000, or 5 times 10 to the power 4.